This video is an excerpt of our Fall 2024 update video. This discusses the many world firsts that DSTRI.org was able to accomplish over the summer of 2024. Thanks for watching. Our second dig this spring and summer was in Montana again at Glendive in the Hell Creek. We spoke earlier of our finding of nematodes after Kirsten Peterson's project. We dug many soil samples. One nematode was found and collected from soil samples. One nematode was also collected from the fruit trap method, which we discussed earlier. But we actually, because of the poor results we were getting from the soil and from the fruit, we decided to extract a freshly dug bone shard from the location where the Triceratops horn was located and where other bones have been collected from that site. We put that bone shard into a Bowerman funnel and waited a few hours, and we actually collected a worm that came out of that bone. And here's a nematode that was collected from a bone shard. This is another world first discovery. It's a preliminary discovery. We had dissecting microscopes with us at the dig to check our work with the Bowerman funnels, but we had no attachment camera, so we were able to use this cell phone to videotape through the eyepiece, and you can clearly see this moving worm. This could have only come from the bone, which was thoroughly cleaned before it was placed in the funnel. And so this is a world first discovery, a living organism, a living nematode inside a dinosaur bone. This was later identified by an expert at Virginia Tech as a fungal-eating nematode, which is what we expect because we found so many fungal infestations deep within these bones. So this is a world first. It does need to be corroborated. More specimens need to be captured and sent to the experts in Virginia for identification, but we're very excited about this world first discovery. We need to corroborate this over and over again, which we intend to try to do. But after we filmed this phone cell video, I was able to collect that nematode from our specimen container, put it into fixative, and transported it back to the DSTRI lab, where we were able to photograph it under high magnification using optical staining methods like Nomarski, as you see here. And this was identified by a scientist, a PhD scientist at Virginia Tech. He identified it as from the Apellicaida, I guess that's the way it's pronounced, Apellicaida, and that's a fungiferous or fungus-eating nematode, which is what we expected based on our discovery of fungal fibers, fungal hyphae growing throughout these bones, particularly in Montosaurus. And so we're very excited about this identification of a fungiferous worm, a fungus-eating worm, and so much more work has to be done on this. But this is another world first that we're really excited about, and we're hoping to capitalize on this as well. We're also finding very interestingly tiny insect remains, microscopic jaws, and also bodies, complete bodies, and other insect parts in our decalcification solutions. And so we know that insects are entering these bones, even tiny ones are getting in there and predating upon the soft tissue and fungus and bacteria that are living inside of these bones. And so that's another world first. We can't find any examples of insect parts in the literature in dinosaur bones. There are quite a few papers on the activities of insects and other organisms on these bones and the way they leave the remains with burrowing tunnels and jaw marks and things like that. But we've never seen anything in the literature that shows insect jaws and full bodies of insects. So we think that's another world first.